All right. So we talked about mRNA. We talked about what it's made of. We talked about the codons. We talked about tRNA. We talked about what it's made of. We talked about the anticodons. So now we're going to talk about some ribosomes. And ribosomes, the structure of ribosomes is kind of interesting. They have two units, a large and a small. The large has binding sites for tRNA. Those are spots where tRNAs will come in. We'll see how that works in a second. And then the small kind of clamps the mRNA into place so that it can be read. And between those two pieces, that's going to give you all of the parts uh, for your ribosome. So here is your ribosome. There's your large subunit. There's your binding site. This is an a tRNA. This tRNA is matching the AUG. Oh, I remember AUG. AUG is the start, right? And I know the start is methionine. You don't have to know it, but I just know it because I've seen this more than once, right? So AUG matches with UAC. A goes with U goes with A, G with C. And then every tRNA that's UAC has a MET. All right. So this is in place. The MET's in place. The start's in place. The ribosome now knows it has to read these every three. It read it read these three, and then these three, and then these three, and then these three, until it got to the AUG. And once it got to the AUG, it's like, okay, now I don't need to read these by ones. I can read these three at a time now and not look for the start anymore. I found my frame. My reading frame is set. Okay. The next one that's going to come in is leucine, right? This incoming tRNA, and it's going to match this, right? Which is CUU. And I'm figuring it out by the colors, okay? This is orange, that's C, brown, brown, UU. So if that's CUU, right? Then this is G, right? AA. Because U and U go with A and A, and C goes with G. So this one's going to slide in to incoming tRNA. It's going to slide in because it's the next one that matches the codon. And there it is. And what you do is you shift the methionine over, you shift it over, and then this ribosome slides forward by 3. And then this tRNA can fly off and find another methionine. Where does it find it? In the food you eat. Whenever you eat food that has protein in it, and this is why it's important that you have a healthy diet, is to be able to get your amino acids you need, including methionine. Then this would be the where you'd find it. So here's your met, and then Lu, and then this whole thing's going to shift to the right, and the next one's going to match UGC. UGC. So that's the codon. The next codon is UGC. So this should be AG. Sorry, whatever it would match with. Okay, I don't have the letters in front of me, and I forgot the colors. But whatever these are, the next anticodon has to match them, and it's going to bring its own amino acid. So here comes the next one. Right. So the anticodon for AUG is coming off. It's going to find another MET. Here comes the next one. It's coming in. And then this chain is going to attach to the cis. And it's going to keep doing that until you get to a stop codon. When it gets to a stop codon, the ribosome, well, stops, detaches. The small subunit and the large subunit separate, freeing the mRNA. And then the ribosome can go attach to another mRNA and make more proteins and more amino acids, et cetera, et cetera. And this continues, right? You could have multiple ribosomes on the same mRNA making proteins at the same time. But the idea is every time you bring a new piece in, the chain shifts over, you bring a new piece in, the chain shifts over, the ribosome moves 3 at a time, and you keep doing it until your protein is made. And that's translation, because you're going from, when you went from DNA to RNA, DNA was made in nucleic acids, right? Nucleotides. RNA is in nucleotides. Even though there's used different. That one nucleotide is different, but it's the same. So it's called transcription. But this is translation because here you're going from nucleic acids, right, nucleotides to amino acids, which is a different language, if you will, right? So that is translation. And that is it for the incredibly long Chapter 8, Section 5.